What are the basic building blocks of DNA and RNA? The correct answer is C, nucleotides. And each nucleotide consists of a 5-carbon sugar, a phosphate group, and a nitrogenous base. And this nitrogenous base can be adenine or guanine, cytosine or thymine or uracil. Section of DNA has 27% thymine. How much cytosine will it have? The correct answer is A, 23%, because we used Chargaff's rule, in which the DNA, the amount of adenine equals the amount of thymine. So if thymine is 27%, adenine is also 27%, and together they will give 54%. And then the remaining percentage must be guanine and cytosine, which is 46%. Then divide by 2, you will get the percent of cytosine, 23%, and also for guanine, 23%. Which was a conclusion of Griffith's work with Streptococcus pneumoniae? The correct answer is C. And let's remember together the Griffith's experiment. In the first step, when he injected the mouse with the rough strain, the mouse lives. Then in the second step, when he injected the mouse with the smooth strain, the mouse dies. Then in the third step, when he injected the mouse with the heat-killed smooth strain, the mouse lives. Finally, when he injected the mouse with the rough strain and the heat-killed smooth strain, the mouse dies. Then he concluded that something from the dead smooth strain transformed the harmless rough strain into virulent smooth strain. Which label represents the nitrogenous base? The correct answer is C. What technique did Hershey and Chase use in their experiment that led to the conclusion that DNA, not protein, was the genetic material in viruses? The correct answer is C, radioactive labeling. And let's remember together the experiment. They used radioactive labeling to distinguish between DNA and protein. They labeled the DNA of viruses with radioactive phosphorus and the protein coat with radioactive sulfur. By tracking which element entered the bacterial cell during infection, they confirmed that DNA is the genetic material. With what does the synthesis of a new strand of DNA begin? The correct answer is A, RNA primer. Which is true about the elongation of the lagging strand? The correct answer is B, Okazaki fragments, because the DNA polymerase enzyme can only synthesize in the 5' to 3' direction, and the two strands are antiparallel. The lagging strand must be synthesized in short, discontinuous segments. These kind of segments are called Okazaki fragments. Which describes DNA replication in prokaryotes? The correct answer is B, occurs in two directions. Which catalyzes the addition of nucleotides to a newly forming DNA strand? The correct answer is A, DNA polymerase. And the primary enzyme responsible for reading the template strand and the catalyzing the addition of new complementary DNA nucleotides is the DNA polymerase enzyme. What best describes the DNA molecules produced during the process of semi-conservative replication? The correct answer is A. They contain one strand from parental DNA and one strand of new DNA. Which correctly lists the changes to eukaryotic pre-messenger RNA to form messenger RNA? The correct answer is C. What is the messenger RNA sequence for the template strand DNA sequence in the figure? The correct answer is B, because according to the base pairing rule, A binds with U, T with A, C with G, G with C. But if we are making a DNA strand, we are not using U or uracil, we are using thymine. How many messenger RNA or DNA bases make up a codon? The correct answer is C, 3. What is a codon? A codon is a 3-nucleotide sequence in messenger RNA that specifies a particular amino acid during protein synthesis. For example, the codon UUC codes for the amino acid phenylalanine, which transports amino acids to the ribosome. The correct answer is C, transfer RNA, which is not a type of mutation. Of course, C, RNA interference. And it's a cellular mechanism for regulating gene expression by silencing or degrading the messenger RNA. Which is true about prokaryotic gene regulation? The correct answer is C. Operones often control transcription of genes. Which demonstrates an insertion mutation of the sequence? The correct answer is D. What do Hox genes regulate? The correct answer is D, differentiation of cells.